we are back with another last minute advice video for A-level biology on paper 2. Listen, I don't care how well or how bad or how mid you did in paper 1. Yeah, put that at the door. We're not talking about her anymore. She's finished. All we can focus on is paper 2. And hot take, I think paper 2 is so easy. Of course, I can only speak for OCRA, really, because that's the example I took, but it's actually so easy because you just need to know the content and there's some level exam technique required, but genuinely, if you know the content, you're so solid. So you understand what I mean as we progress on this video. But as always, got 3 stars last year, know what I'm doing, let's go for it. So mindset, I always talk about mindset because it really does matter, because I know loads of people who after paper 1 felt so demoralised because questions were weird, paper was weird, the vibe was weird, whatever, but some people thought it went really well. Again, it's a mixed bag, but it doesn't matter. It is your time to lock in, and everybody knows that we're all locked in. However, you need to lock in efficiently. So the steps that I do tell you are very specific to paper 2. This order that I've done it for is not going to be the same for paper 1 or the next paper 3 last minute advice video I'm going to make. Very much specific to paper 2. You see, I know for a fact you guys look like that. Like, I can bet money on it that you guys look like this. You're like, oh, flip, another all-nighter ahead of me. I'm so sick and tired. When is I don't know, let's say your final exam is chemistry, so paper 3, that's I think 21st of June. You guys are like, when is the 21st gonna come? Don't worry, it's gonna come, it's fine, but hopefully after the video you feel like, yeah, I know what I'm doing, this is gonna be great. Okay, that's the vibe we're going for. So lock in efficiently, and here is what I would suggest. Okay, we've got a total of 3 steps. And I'm going to go through some exam technique stuff like I did with paper 1. Loads of you guys said that evaluation questions really that I went through in that video helped you guys so much, and I'm so happy to hear that. Right, if you're an OG, OG since May, you guys know that normally I tell you to speedrun content as the last step. But I'm not telling you to do it as a last step. I'm telling you as the first step. And I have a very specific reason why. Again, I can really only speak for OCRA, so take what I say with a pinch of salt if you take another exam board. But when I tell you paper 2 is so much content, it's just explain this, describe this, outline this, it's actually a walk in the park. It's so easy. Don't come at me in the comment section, please, okay? But the questions are genuinely orientated based on content. With paper 1, there's so much evaluation. Gosh, what can you conclude about this scientist's statement? Evaluate this claim. Give me a break, yeah? And the break is in paper 2, because it's just explain and describe, really. So when I tell you, you guys need to look into your content, I mean it on a different, frequency, yeah? This is for OCRA. Let's just take a little look, right? Paper 2 is assessing module 1, 2, 4, and 6. Module 4 and 6, let's just have a look, right? Module 4 is biodiversity, evolution, disease. Module 6 is genetics, evolution, and ecosystems. When I tell you, I can guarantee that there will be lots of marks on sampling, biodiversity, ecosystems. That's part of the content that's going to be assessed, but they love that. They love it. Listen, OCR loves sampling because barely anybody can really do those questions. Let's be real, who really listened on the field trips when we were doing ecology? Yeah, silence. Anyways, so you really need to make sure that you know how to do sampling methods. Let's say with genetics, how to do patterns of inheritance, right? Knowing what dihybrid inheritance is, how to do Punnett squares, how to do stat set calculations, e.g. height. What's it called? I've forgotten now. You know what I mean? It slipped out of my tongue, but Hardy-Weinberg, that's the one, yes. Literally just recorded a video on it yesterday, gosh. Yeah, also in terms of cloning and biotechnology, OCRA, gosh. When I tell you cloning and biotechnology is there, like, it's their queen, it's their king, it's their everything. They live and breathe for that topic. Every year there's questions on cloning and biotechnology, especially serial dilutions, every year. Because it's kind of hard, you know, there's lots of level of understanding required for it. Cloning, all aspects of it really, but aseptic techniques comes every time. So that's why I'm telling you, literally, you speedrun that content. I don't care if you've done any questions or not. I actually do not care. What I care is that if I tell you, explain PCR to me, if I tell you, oh, fill in this like Punnett square for sex linkage and whatever, you will be able to do that. That is the level I need you to be at. Because genuinely, if you have no exam technique, you'll still pass. But imagine you know all the content and then you have exam technique. Oh my gosh, a match made in heaven. So, I mean, yeah, use YouTube. Honestly, don't bother making notes. If you use flashcards, go ahead, Quizlet. Sets are pre-made or Anki, whatever. But me, personally, I would use YouTube. I don't have time to be doing flashcards. No thanks. Right. This section is kind of funny. I bet you weren't expecting a picture of Nick Wilde, if you know where he's from, the movie Zootopia, but I have a reason. Yeah, I actually have a valid reason. So step 2 is all about exam technique. Now before we start that, again, mindset. Like when I tell you your mindset is your make or break, because I've been through the thick of it last year, I'm aware that your mind could be a hindrance or it could be your pusher. But when you answer the questions, you need to imagine the examiner is a 7-year-old. I've always said this and I'll say it again. A 7-year-old does not care for flowery language. A 7-year-old is like, tell me the facts now. Our age, people, you know, nowadays, beating around the bush. Relax, we don't have time. When you write your answers, you're going to go like bish, bash, bosh, like state all the obvious points, just say it how it is. And that comes with a bit of practice, because I'm saying this because it's so easy to overthink questions. Like, you read the question, you're like, oh, I think I know what to do. And then when you get to the answer and you finish writing it, you read it, you know, over, and you're like, so did I even answer the question? You don't even know, do you? So just read the question, rephrase it, see what topic it is, bish, bash, bosh, write your answer, move on, call it a day. So imagine the examiner is a 7-year-old and keep repeating that to yourselves. So, as I said here, it means that you should answer the questions in the most point-blank period way. But how would you do this? Be like Nick Wilde, okay? This character, I thought about it, right? And I thought, what's the best way to instill this into you guys? And I thought, Nick Wilde is the best example. You know when sometimes hard situations occur in life and you perhaps think, oh, what would this person do? Or like, what would this character in this movie or series do? Like, you know, you're trying to gain strength or bigger perspective. Well, you can do the same here. So if you don't know who Nick Wilde is, you're living under a rock from the best movie ever, Zootopia. But there's something really important I want to talk about. His qualities are that he's sly, wise, and street smart, and slick, really. So when you're answering questions you're overthinking, just be like, I need to be like Nick Wilde. He has such swag to him. It's crazy. And he just kind of cruises through the movie. You cruise through the paper as well, right? They're a 7-year-old. Be slick about it, be street smart about it. They're asking about aseptic techniques. You talk about, preventing contamination. We want, I don't know, like, but etc. You get the point, right? So the things that you should stop doing is overthinking, not simplifying the questions, and not answering the actual question at hand. They're the things like, get that out the door. We do not want to be doing that. So to prevent this is try and embody Nick Wilde. And I kind of highly recommend watching a Nick Wilde edit on YouTube. You'll understand what I mean by these qualities. So let's try and, embody his qualities as such to make sense of the sticky note over here. But before we actually do that, strategies. Just assess how do you want to answer the paper, right? Personally, I skip multiple choice. I'm like, not today. Don't want to do it. It just takes up too much energy. So I go straight to the written questions. After I finish that, of course, I'm skipping hard questions or questions that I'm like, I don't really know what to write. Star it, move on, call it a day, come back at it, at the end, after I've done the multiple choice questions, and then, go through the skipped and hard questions. And then the next thing is like, what's your personal exam technique strategy? So mine is reading the question first, squared, twice. Then I'm going to rephrase it into something that just makes more sense. Because sometimes I just waffle, and I don't know. I don't have the capacity for that. Then I would check the command words. So if it's explain, I would know the level of detail I need to go into. If it says outline, again, I know the level of detail I need to go into. Of course, I have a playlist all about command words. It's like my first set of videos on the channel, so go have a look at that if you need help. And then see if I need the information about the question, because I just said in my first step I'm reading the question first. That does not mean that I'm reading the stem of the question, right? Like, this is all information and this is the question over here. I don't care about this. I just go straight to the question, because genuinely, like 8 out of 10 times I can just answer it straight away. There's no need for information. Why should I just confuse myself even further? So I just avoid it. But that, again, is a personal method. Try this method if you don't have an exam technique strategy, and adapt it to however suits you. Right, now let's speed run through some exam technique. So I've got a bunch of questions, and actually, just before I go through specific ones, I just want you to see the vibe of it, right? 6 marker, using the figure, explain why some gene mutations do not affect the function of protein. Easy, standard, like content, hello. Oh look, aseptic techniques, precautions, state why it's important, easy, content. This, serial dilution's respectable, quite hard, but if you know the method you'll be fine. I've got a video going through serial dilution questions on my channel. I think it's like from 2 months ago, I think. So go have a look at that if you're still struggling. Suggest why they shook the flask, suggest one reason why they're difficult to count, like, oh, come on, these are easy marks if you just know aseptic techniques, right? That subtopic. Here, suggest explanations for the changes, easy stuff. If it's increasing, then like, what does that mean? When is it increasing? Is the certain population decreasing at that time? And then lastly, 6 mark again, selective breeding, maintain viable wild population. Easy. You'd learn that in module 6. So hopefully you kind of realise what I meant at the start when I said that, yeah, paper 2 is just very much content based. Like, you're not really catch and evaluate question to the levels that there are in paper 1 or paper 3. Paper 2 is just that, like, in between, like that little gateway from paper 1 to 3, and it's just so easy once you know the content. So let's just speed run through this. So read the question first, so use figure 16.3, and okay, if you don't say both, like if you only talk about protein structure but not the figure, then your marks are capped at a certain level, so just make sure you realise that. Explain why some gene mutations do not affect the function of a protein. Honestly, I don't need to rephrase that. It does the job, So let's just have a look at 16.3. Right. Essentially, this is the genetic code. So it's saying, for example, if you have G and you have C and you have a combination of, let's say, GCG, GCA, GCC, GCU, it's going to produce alanine, right? This means that the genetic code is degenerate, so multiple codons can code for the same amino acid. So that links to why some gene mutations do not affect the function of a protein, because it is degenerate. So that means the primary structure won't be changed, the tertiary structure won't be changed. Some mutations are silent, right? It would again leave the primary structure unchanged. So just take a moment with your figures and your tables to try and understand what it is. And if you're ever stuck, just think, what topic is this? And keep narrowing it down to the subtopic. And then that's how the mark scheme reflects it, okay? Now, right, state why it is important that the technique used for culturing microorganisms be aseptic. prevent contamination. I didn't read anything, like it's just so obvious. Plus it's state. State means like just say how it is. You know it, okay? Yeah, easy. And then second, list 2 precautions that should be taken to prepare a culture to make it aseptic. Okay, think back to your practical that you did in class, right? Oh, I don't know, disinfect surfaces, work near a Bunsen burner, flame. Pretty good, isn't it? As I said, it's all content. Next one, suggest why the student shook the flask. You know, I didn't screenshot that, it's fine. You know, you want to distribute the bacteria across the solutions. Suggest why it's difficult to count. Oh, well, you know, they're too small, there's too many, stuff like that. Now, onto this question over here. People do not like zero dilutions, I'll tell you that. But this question is quite different, I'll be honest. So I did go through it. Basically, so in one 0.01 centimeter cube sample, the student counted 52 bacterial cells. I always draw a diagram, yeah, because it just makes more sense. So that means if they're counting 52, you're starting from here. You're basically going to be working backwards. So you have 52 cells, and it says describe the calculation steps the student would take to estimate the total number of bacteria in the small flask, basically the original. So always look at what information is said, you know, prior to this question. So we know they're doing it by 0.1 centimeters cubed and 9.9 centimeters cubed of water. We also know that the number of small flasks each contain 50 centimeters cubed of bacterial culture. So this, and this are the 3 pieces of information we need to use to calculate this question. Basically, as I said before, you want to start backwards. So if you have 52 cells and it's like they'd taken a sample that was like 0.01 centimeters cubed. So you need to scale that to 10 centimeters cubed because that is the total of this like test tube. So to do that, you need to times by 1,000, right? Those cells times by 1,000. Now, the thing is, we don't actually know how many times we're diluting it. But for a fact is that we are taking out 0.1 centimeters cubed each time, right? Now, typically, it's not actually 0.1 centimeters cubed. Well, normally, like in other questions, it's actually 1 centimeters cubed. And in this case, it'll be times 10. But the fact that we're taking out 0.1 means that we times by 100. So because we don't know how many times we're diluting it, we can just do 100 to the power of n, which n is just the number of zero dilutions. And then, well, we basically, ellipsis, we go through all the test tubes for how many times you're diluting it, and then you arrive at the original. Now, we knew that it was at 50 centimeters cubed, so we can times by 5 because of the scaling that we did with the times by 100. Like, if you didn't do, I think they said it here, didn't they? Yeah, wait, let me check this. No, not that one, this. If at the start, over here, you didn't times by 1,000, but you did 100, right? If this was 100, then this becomes 50, okay? It's a bit techie, I know, but that's kind of how you do it. So just take a moment for this mark scheme, okay? Cool. Again, there's other videos that I've made about zero dilution, so go have a look at that if you're still confused. Oh, right. Next one. Okay. Suggest explanations for the changes in the population of chinstrap penguins between 1976 and 2010. Okay. Number 1, identify where the chinstrap graph is, so I've highlighted it. And it says explanations, right? So at this point, you just need to see, like, what's happening at each stage. So if it's increasing over here, oh, well, how ironic. The Adelaide population is decreasing, okay? Also, how ironic here. It's kind of decreasing over here, but at the same time, the Gentoo population is increasing. So just think back to the topic, right? About, like, ecosystems, population sustainability stuff, and think, oh, you know, niches, competition. Oh, competitive exclusion principle is something I want you to search up. This is always kind of like a mark with this topic. For OCI, I don't know about like AQA and stuff, but yeah, competition, food resources, and just that, you can be specific. Who's arrived that's led to them like decreasing. As we said, Gen 2 is increasing at the time it's decreasing. This is all, they don't put a graph. That doesn't make sense, right? So, yeah. Again, explanations, simply content answers. Then lastly, another 6-marker, explain with reference to selective breeding why it is important to maintain viable wild populations of crop plant species. Okay. If you don't know this, genuinely just like memorize these points. But essentially, you want to maintain viable wild populations. That's like the OG plant species of a crop. So, of course, selective breeding, genetic engineering and stuff has occurred. So that means that it's not wild anymore. It's like GM, right? Genetically modified. Wild is like the OG, as I said. So they're saying like, oh, why is it actually important to keep this? Well, you need genetic variation. If you're doing genetic breeding, genetic engineering and selective breeding on plants or crops, well then you're decreasing the gene pool and stuff. You're selecting for certain characteristics. And so what if environmental changes occur so much that like all the crops are dead? What's going to happen then? So that's why we need genetic variation. And linking to that, we store it in a gene bank. If you don't know what that is, search up. It's actually really cool. Again, source of useful alleles, like they've been crossbred. Potentially useful in changing climate, because environments change. So if every crop is basically the same, well, they're not going to withstand the changes, are they? You don't want inbreeding depression at all. You want this. For some reason, nobody really. Well, okay, I don't want to overgeneralize, yeah, but not many people know what hybrid Viger is. It's just weird. Like, if you don't know what it is, please search it up. If you go onto Google Images, you'll clearly see what this means. Chuck it in, about when it comes to like selective breeding and stuff. And yeah, that's pretty much it. Again, content. I picked these questions. I mean, they're just from I think across two papers, but genuinely, like, I'm the exam technique person. I tell everyone you need to sort out your exam technique, But the first time in my life, I'm telling you to focus on your content, because genuinely, if you know your content, you can just bash these out. Simple, very easy. Okay, which then leads to my third point. Make your frameworks for each topic alongside doing past papers. Okay, yeah, you need to learn the content, but test whether you've memorized it properly. So use past papers for that. So these are literally just a few things, okay, that I just wrote. So like cloning and biotechnology, if I get a graph, like bacterial population growth graph, then for some reason I keep forgetting, I forgot to talk about these things before, like birth rate's bigger than death rate, So I just wrote them down for myself. Just like other things I wrote down, just to remember, like underneath a topic, natural selection. Let's be real, the answers are literally the same every time. So that's why I say make your frameworks. If you're doing a little mini notebook, then bonus points to you, if you get the reference. But these were like 2 sticky notes I had before the exam for paper 2, just like, well, it's actually mixed with paper 1 and 3, but it doesn't matter. Just to remember. Again, frameworks help you to remember stuff, and ultimately that's where the marks come from. Okay, these are like mark scheme stuff. So yeah, just go through each topic that you do, like let's say module 6, do some past papers, do some topic questions, and if there's certain things that keep coming up or that you should talk about for certain topics or top topics, write it down. Yeah, cheat the system. This is all a game. You're a player. Like, play the game, right? So, and lastly, really, as I said, doesn't really matter how you did in paper 1. All that really does matter is that you keep going and keep getting marks. This is all a game. Please just be as calm as possible, right? You probably will work better when you're calm than stressed. And hopefully, after the paper, you're like, yes, I cooked that. I did that solid, you know, moving on to the next paper. And just remember why you're doing all of this. Of course, we were all forced to do A-level exams, like who wants to do them? But at the same time, this is your dream. Most likely you'll need these grades to go on to the next step. Or maybe you don't. Maybe you have this like dream university that you need certain grades for, right? This is your dream. Like, not to put pressure on you at all, but just like you have a purpose as to why you're doing these exams. A bigger purpose than you probably think right now, stressing or pulling up like all-nighters. But honestly, this is all going to be finished in 2 weeks, and you're not going to think about it again until results day. But you know, who cares about that? That's in a few months. Yeah, just work for your dream. Yeah, nobody's going to work for you. This is all a game. Just act as the player and cheat the system, as I always say. And I really, really, really do hope that these steps were useful, and I hope paper 2 goes swimmingly well for you. And yeah, I just hope you become like this. Yeah, as always, write down in the comment section like what your plan is for paper 2, any tips, any advice you have, like for everyone, you know, collaborative space and all of that. If there's any certain videos that you guys want to see as well, also write it down. I'll try my best to do it. And very best of luck, and update me on how paper 2 goes. See you guys, bye!